Hi everyone, good morning. I hope that everyone's doing well. I'm Fasika and today Varman and I are going to present on the topic SOD1 is a possible predictor of COVID-19 progression as revealed by plasma proteomics. This paper is done by the following people presented below. Now moving on to the introduction, what exactly is coronavirus? Well, it is a disease that is caused by the severe acute respiratory distress syndrome associated coronavirus 2 and is considered as a threat to the whole world population. It is also declared as a global pandemic by World Health Organization on March 2020. As of 16 March 2021, CDC has reported that almost 120 million of the world population are infected by coronavirus and more than 2 million people have already lost their lives. So what are the symptoms of this disease? It's very basic: fever, cough, and fatigue. And the widely used method for the detection of this virus is chest radiography and reverse transcriptase PCR. However, these methods fail when it comes to revealing the molecular mechanism or the prediction of disease progression. As we know that proteomics has been widely used to identify biomarkers and disease progression for various infectious diseases. Even in this disease, the serum biomarkers from the patients with severe respiratory syndrome is identified by proteomics. Lately, it has been accepted that plasma can also be used to reflect pathophysiological alterations in progression of this disease, whereas COVID-19 are classified as asymptomatic, mild symptoms, or severe symptoms based on their clinical status. Prediction of different COVID-19 outcomes, dysregulation of macrophages, platelet degranulation, and complement system pathways were detected in COVID-19 patients by the help of proteomics. These studies have broadened the knowledge of plasma proteome and metabolite changes in COVID-19 patients. Despite several studies, still the treatment of coronavirus and predictions related to this disease is limited. The main aim of this study is to investigate the proteome changes in COVID-19 patients with asymptomatic infection, those with mild syndrome, and severe syndrome in the early as well as the later phase. Also to identify the changes that occurs in plasma proteins by the COVID-19. And finally, can superoxide dismutase 1 be used to predict COVID-19 progression? Moving on to the materials, all the reagents mentioned were used for the methods below. Firstly, plasma samples were collected from patients with symptomatic infection, severe syndrome in early phase and in later phase, and healthy controls. All these experiments were performed in Biosafety Laboratory 3 facilities. High abundance proteins were isolated and plasma protein concentration was detected, followed by TMT leveling, where each plasma sample was processed for TMT leveling study. Protein samples were reduced, digested with trypsin, and desalted using reversed phase Collin chromatography. 6 sets of samples were subsequently separated into fractions using high-performance liquid chromatography. Fractions were collected every 1.5 minutes in 45 tubes, dried, and again combined into 15 tubes for further liquid chromatography tandem mass spectrometry analysis. Peptide Fang fractions were analyzed using a mass spectrometer. All the protein abundances were subjected for further bioinformatic analysis, protein abundance normalization, subsequent heat map, and volcanoplot analysis. Then, gene set enrichment analysis was performed to identify classes of genes, phenotypes, and statistical significance. ELISA was also performed in Biosafety Laboratory 3 . followed by statistical analysis to evaluate the differences between each disease status group and also healthy control group.
So talking about the results, we got from the analysis. So the first result was like there was various changes recorded in the plasma proteins of COVID infection patients. So to investigate this, about 630 plasma proteins were collected from COVID-19 patients. Also, the SC group members were being recognized, which were less than 1%. They analyzed about like 7,944 group of peptides, which contain like 120 proteins which were dysregulated. So talking about different groups like MS, SSCP or SSO groups, which had 61, 48, and 49 dysregulated proteins respectively. But as comparing to SC group, there were 17 group proteins which were downregulated, and 2 proteins like gelsin or genus kinase were upregulated. So we can check go the results regarding the dysregulated proteins, which is being mentioned related to the different groups in the table provided in the article. So I will provide the link for the article so you can have a look at the table as well. So there are 4 figures showing different number of people landscape of the plasma proteome being analyzed, what changes the landscape get during the COVID infections. So moving on to the next result, which was being performed for recognizing the concentration of dysregulated proteins in different cellular parts. The analysis is called geoanalysis. This geoanalysis recognize all the cellular components of the cell to find out which is enriched with the dysregulated proteins. The cellular components, especially like extracellular exosomes, blood microparticles, or extracellular regions, were being investigated, and the result was found that they were being enriched with dysregulated proteins. Different binding proteins such as cadherin, which act in cell-to-cell adhesion or acting binding site or serine type endopeptidase, were also enriched with the dysregulated proteins in the COVID patient of different groups such as NS, MS, and SSL groups. All the enrichment of the dysregulated proteins are being shown in the figures like ABCDD. So you can have a look at that. It will make you clear about the different concentration of the dysregulated proteins in different cellular components. Our next result was finding out the relationship between the dysregulated proteins and the PPI system. So it was found out like the dysregulated protein showed a very close relation with the PPIs, but some of the functions like coagulation cascades, which contains glycolysis or glycogenesis, were dysregulated in different groups such as NS groups. But some groups like MS group have the cholesterol metazollable proteins which were downregulated due to the COVID infections, which caused the dysregulation in their protein system, including various systems which is being mentioned in the article, which is APOA, APOA2, APOC, APOM, LCATA. But it is found out like the more proteins were dysregulated in the SSLP name groups, and various functions such as coagulation cascades and streptophilus aureus infections. So all the dysregulation proteins link was being found with the PPI through the KEGG pathway, which is being shown in the figure provided in the PowerPoint slide. In the next result, SOD1 was considered as an important marking point for the recognizing the severity of the COVID-19, like indicating the harmness of the disease. It was being done by GSC of the dysregulated proteins, as shown in the figures. About there was being performed in different groups like MS, SC, or SSP versus SC or SSP with MS. So for performing the verification of expression of the protein in the cells, we perform the ELISA for three proteins, SOD1, LDAH, and PRDX2. It was being mentioned in the article which was like PRDX2 and LDH levels were really high in the SS group, but not in the other groups. The figures show the different levels of the protein expressions. SOD1 protein also used to distinguish between the patients which are being infected with the SARS-CoV-19 virus from the other normal groups such as SC groups. So by using SOD1, we can distinguish between different groups, which is being shown in different figures. So you can have a look at that. For the discussion part, like they use the TMT labeling for analyzing the relation between the proteomic changes with the changes being performed in the clinical labs of SARS-CoV infection patients. Like they also mentioned, like age was one of the important factor for determining the harmness of the COVID-19 disease. It was analyzed that the average age in each group was different, so it's hard to identify the certainty of harmness of the SARS-CoV infection. Oxidative stress, blood glucose level, or ENO1 also played an important role in determining the severity of the SARS-CoV infection, because if there is a COVID infection patient, so it will show oxidative stress, which is struggling to have the oxygen. So blood glucose level like also played an important role along with the oxygen level. Also they provided the complement activation which is used to being treat the COVID patients, but it is really hard to imply the controlled complement activation, as it may get the undesired results. There were also some limitations regarding the analysis, such as they didn't go the groups with no age difference, which was like if there is a age difference between the different groups, it will limit the study of the analysis of the COVID infection patients. Also, as mentioned earlier, oxidative stress and COVID-19 disease were interrelated, but there were no specified information about it, and patient numbers also not enough to be analyzed for the study, and the study remained limited. And as I mentioned, a treatment of the COVID-19 is complement activation, which is really delicate to use because it can harm the life of the patient. So as a conclusion, we concluded that according to the given article, a patient with COVID infection faces the changes of the protein particle present in the cell, which is it changes the landscape of the protein morphology and various other effects. Also, oxidative stress and SOD are the key points to determine the harmness of the disease, and the levels are up-regulated or down-regulated. So they make the COVID infection in the patient. Also, the pathways used for the treatment of COVID-19, such as complement activation, is really harmful to use, but it can be used to some limited extent. So that's our end of the presentation. Thank you all for listening it, and if you have any questions regarding it, you are free to ask. Thank you.